the cilium of eukaryotic cells beats with a characteristic stroke that goes like this. There's a power stroke in which the cilium is straight, and then a recovery stroke in which a bend propagates along the cilium so that the recovery stroke undercuts the power stroke. And swimming, that is the movement of water, so that the cell either moves water past itself or the cell moves through the water, same thing, is fundamentally accomplished by the difference between this and this. If we take out a little piece of that, dragging this cylinder, this cylindrical segment through the water this way, experiences a lot more drag than dragging it through the water this way. Now, in any discussion of the swimming of cells, inevitably one encounters the following physical concept of Reynolds number, always symbolized RE, R little e. Reynolds number is a dimensionless ratio. Forget about how we arrive at it, but it approximately means the ratio of inertial forces to viscous forces or drag forces. In the numerator appears the square of the velocity, the square of the length, and the density of the medium through which this body is moving. That's density there, Greek letter rho. In the denominator, we have velocity, length, and viscosity. Of course, viscosity is the dominating phenomenon for drag-based fluid motion. Now, obviously, the L's cancel, one of the V's cancels, so you're left with speed times length times density. Now, we're talking about water all the time in this course, so let's just say the density stays the same over viscosity. The viscosity doesn't always stay exactly the same. Okay, there's mucus, there's things that affect the viscosity of water, but at any rate, the point is this number, the ratio of inertial forces to viscous forces, is large when things are moving fast and when those things themselves are large. If Reynolds number is 10 to the third, you can safely ignore drag forces, the forces due to the viscosity of water. For something like anything larger than a goldfish, that's the case. For a tuna, Reynolds number is probably on the order of 10 to the fifth or 10 to the sixth. What this means functionally is that a tuna swims by throwing water around. As it waves its tail back and forth, it's accelerating blobs of water that impart inertia to the tuna's body. In contrast, for most cells, a cell on the order of 10 microns or something Reynolds number might be on the order of 10 to the negative third or 10 to the negative fourth. In other words, you can ignore inertia. All of its motion is dominated by viscous forces. There's an interesting middle regime where they both matter, and in general, biologists don't know how to deal with that, so we like to make sure that we're way away from one. Okay, now for a cilium, what this means practically is that a cilium can't throw water. It's only able to drag water around, okay? If there's no inertia, then as soon as the motor stops, the cilium stops. Now let's talk about what's inside a cilium. Let's say the cilium borne by this epithelial cell. If we blow it up, bear with me, I'm going to draw first the basal body and the axoneme that grows from it. We'll look at what's inside this thing called the axoneme, the skeleton of the cilium, in a moment. The basal body is associated with some appendages, which in turn are associated with what are called microtubular roots, ribbons of microtubules that extend down into the body of the cell. Microtubular roots are, on the one hand, a structural support anchoring the basal body and its axoneme to the nucleus and the rest of the cell body through cell junctions that I haven't drawn in this diagram. But they are also organizers for traffic in the cell, typically. There's another structure attached to the basal body closely in most cells that have a beating cilium. This thing is called the striated rootlet, and it includes a number of proteins. No one is really entirely sure in all cases what it does. One of the proteins that includes is a small calcium binding protein called centrin. Centrin is one of the set of universal eukaryotic proteins. Highly conserved, it happens to change shape abruptly when it binds to calcium, and in at least some cases, this striated rootlet is contractile and can orient the cilium, change the ciliary beat. Centrin makes a number of other structures. In some flagellates, centrin makes a connecting fiber between basal bodies, each of which sprouts its own cilium, and the contraction of centrin will twist the angle of the flagella relative to each other and change the swimming. That's not a feature of animal cells, so I'm not drawing such a thing in here. Now, let's get a membrane on here. That's enough of one for this purpose. It is typically the case that the immediate vicinity of the cilium experiences a higher rate of endo and exocytosis. And somehow the base of the cilium is also associated with a variably complex apparatus called the transition zone that might include spokes or other complicated, poorly understood morphological entities. The general idea is that this specialized area of membrane in the transition zone of the cilium perform a function similar to the nuclear pore complex in gating traffic into and out of the cilium. As we'll discuss later, within the cilium, there are other objects, trains of motors, for example, carrying cargoes back and forth up and down the cilium, for example, to build, maintain the cilium, or other structures within the cilium. Many cilia are highly specialized for sensory functions, for example. So that completes the basic skeleton that's relevant to ciliary function. I want to point out that on some epithelial cell, that cilium might be in the airway where its main job is to move mucus along the surface. That might be 5 to 10 microns long. On a starfish or a sea urchin larva, that might be 20 to 30 microns long. And in both cases, the beat is like the one that I've drawn here. That is a simple ciliary beat. On some other cells, like, say, sperm, the cilium might be very long. It might be hundreds of microns long, even 1,000 or so, and it might beat with a flagellar waveform. All right, let's take a look at now a cross-section of the axoneme. In all motile cilia that I know of, except one, there's a central pair of microtubules. Then around that central pair are 9 doublet microtubules, arranged as if on spokes. I should draw these spokes in a different color because I'm using yellow here as my code for microtubules, and these links are not made by microtubules. So let me revise that quickly. The links are made by a variety of proteins we won't talk about. This structure, this so-called nine plus 2 composition of the ciliary axoneme was first revealed in one of the early triumphs of biological electron microscopy. Electron microscopes were invented sometime, I don't remember, in the 1930s and 40s or something like that. And among the first biologists to really adopt electron microscopy as a tool was the phycologist Irene Manton, who did pioneering work in both cell ultrastructure and taxonomy. And she first observed this formula in the sperm cells of green algae and ferns. And it turned out to be a universal, almost universal trait for the motile eukaryotic cilium. There are some modifications to it. Non-motile cilia typically don't have the central pair. And there are a few other formulas out there for cilia. These doublet microtubules consist of one complete microtubule with a set of 13 protofilaments and an incomplete one consisting of 11 protofilaments stapled to the side of, stapled to the wall of the other microtubule by a special group of proteins called the tectins, which we don't need to worry about anymore. The part we do need to talk about is the dyneins that connect adjacent doublets. We encountered dynein before in the context of things like pronuclear migration and chromosome capture. That was cytoplasmic dynein, which was actually discovered after ciliary dynein. Ciliary dynein was first discovered in the sperm of sea urchins. There are actually two sets of dyneins in the ciliary axoneme. There's so-called outer arm dyneins and the inner arm. My imperfect subdivision of the circle here accidentally illustrates an architectural problem with the cilium, which somehow it solves quite readily under normal circumstances. That, of course, the spacing of these doublets must be such that the dyneins reaching from one to the next must be able to actually span the gap between them. And of course, one assumes that there are rulers in here that space the adjacent doublets appropriately. Dynein in the cilium is arranged in this clockwise fashion. In fact, if you take a cross-section, an electron micrograph of a cilium, you can tell whether you're looking down this way on a cell or that way if you look at the clock orientation of these filaments. Now, the basis for ciliary motion is that dyneins from one doublet engage with the microtubule lattice on an adjacent doublet clockwise from it. And we already said the intrinsic characteristic of dynein is that it crawls towards the minus ends of microtubules. We know the minus ends are going to be down here, anchored at the basal body, and the plus ends, that is the growing ends of the microtubules, are going to be out there at the tip of the axoneme. So I'm going to swing the camera over here and we'll explain the sliding filament mechanism of ciliary motility. Now I'm going to take just a pair of these doublets out. So I need one with a complete wall and one incomplete. Like that. Again, the minus ends are all down here, or in that direction at any rate. And motors along this one are binding to, or at least reaching out toward, this wall. quite obviously, if all these motors crawl that way, then this thing has to slide down that way relative to this one, right? Even to the extent that they might run off the ends of each other. Of course, that's not what's happening in the cilium because these things are connected to a common base. The only way to resolve this, if these things are crawling on each other, one after the other, is for them to bend. Imagine that this happens all around the circumference of the axoneme. What's going to happen to it? Well, obviously, if it happens all around the circumference at once, it's not going to make a ciliary bead. It's going to twist up into a coil, okay? That's not going to work. So this kind of sliding mediated by dynein motors can't be happening simultaneously all around the wall of this engine. It has to go off in some sort of orderly progression. And the secret of ciliary motility is to relate it back to the alternating beat cycle. The secret of ciliary motility is to choreograph both the activity of dyneins along the flagellum in time and also around the axoneme. For example, to propagate a bend one way, the dyneins on this side of the cilium might be active, and then to reverse the stroke the other way, the dyneins on this side of the cilium might be active. It remains a subject of considerable research interest how the structure of the axoneme coordinates the activity of dyneins to produce the ciliary beat. One thing is clear from classic studies around the time of the discovery of dynein, in which people studied demembranated sea urchin sperm and other similar models. You can strip everything away except the cytoskeletal structure of the axoneme, add ATP, and it'll still work. It'll still propagate a beat that looks just like the beat in the intact cell. So there's nothing like axon conduction or calcium pulses or anything like that running up and down the ciliary membrane that organizes this. It must all be intrinsic to this apparatus itself. This model for ciliary function is not the only conceivable model, and it was arrived at through the study of, as so often happens, an especially favorable prep. Peter Satir studied in the 50s and 60s and 70s the gill filaments of mussels, which are so thickly decorated with beating cilia in such great numbers and in such an orderly wave that passes along the gill filaments that he could conduct electron microscopy and know exactly what part of the beat cycle each individual cilium he had cut through might be and where he was along its length. Okay, I'm sure it was hard work, but he managed to do it. And Satir reasoned as follows. If he cut through the tips of cilia, imagine they look something like this. There's going to be a central pair in there somewhere. Now, as it happens, I'm not going to draw this part in carefully here, but the doublets, they're not doublets all the way to the tip. One of those microtubules goes further than the other. I think it's the intact one, the complete one. So he reasoned as follows, that if these fibers, these microtubular fibers, are contracting to make the cilium bend, which was one of the models under discussion at the time, then you should see something like the following. As this thing bends over, make a rather exaggerated bend here, and I'm just going to draw in the ones on this wall. These ought to be the same length they were before, but it ought to be the case that these have in contracting effectively shortened, right? So he went looking for this by electron microscopy. Imagine that. So think about what he's really looking for a moment. If you take sections through cilia and look at them in the electron microscope, it's a very myopic view, okay? So he's not really going to be able to reconstruct this whole thing and say what happens. And gosh, it would be, even for a muscle filament, a lucky plane that happened to go through the whole thing like that. What he's looking for is cross sections of cilia in a known part of their beat. If the filaments were contracting to make them bend, you would predict that you would always cut through these tips at any point in the beat cycle. That you would always get this same 9+2 arrangement. Oh, I hope I got 9. Is that 9? Yes, that's 9. You would always see all 9 of them plus the central pair. What he actually saw in cutting cross sections, going through here like this, is that he saw the 9+2 configuration in cilia that were more or less straight in the straight part of the phase, but in cilia at different parts of their bending cycle, you might find the central pair and a few of those tips on the side facing the bend. Something like that. Now, if you look at the notes, this is drawn out with a little more care in the notes, but it is a useful exercise if you can find yourself some sort of flexible material with which to do so, to assemble yourself something analogous to the axoneme, perhaps out of pipe cleaners or something like that, and try it yourself. That is, in fact, what Peter Satir did in several of his papers. He built physical models of these things. At any rate, this is the original evidence for the sliding filament model. Unambiguous proof came from, not long afterward, from studies of those demembranated sea urchin sperm that I mentioned. So take all the membranes off, and these sperm tails, if you add ATP, and some magnesium, will make a perfectly good flagellar beat. So add ATP, some magnesium, some other things, pH buffer, whatever. That works great. Now, if you gently trypsinize them, trypsin is one of the stomach digestive enzymes. Gentle trypsinization can dissolve some of the links that hold the axoneme together, but it doesn't dissolve the microtubules or the dyneins necessarily. And if this axoneme is, again, composed of the 9+2 filaments, I'm not going to draw all 9 in there, obviously. What Ian Gibbons and his collaborators found is that if they add ATP to these, and whatever else they need, magnesium, then over time, they would see these demembranated axoneme chunks unspooling, uncoiling like that as microtubules slid past one another out of the axoneme. In the absence of the crosslinks that force the axoneme to bend, they'll do exactly this. They'll just slide past each other until the axoneme falls apart.